Welcome back. In this section, we are going to learn about the structural and functional unit of skeletal muscle known as the sarcomere. It gets detailed, it has a lot of terminology. You may have to listen to this twice. You may have to listen to supplemental videos. You may ask for a Zoom meeting with me to go over some details or work together in your study group or have a meeting with a tutor. But it's crucial that you understand this microanatomy before you move on to the next section, which has to do with muscle contraction. And this is some really cool stuff. So let's get started. When we look at a skeletal muscle cell, let's remember that the word sarco, the prefix sarco, also means muscle. So we're going to talk some terminology here when we're talking about these myocytes. And so we're going to change the naming of some of the parts inside the cell. Instead of calling it a plasma membrane, we're going to call it a sarcolemma because the Latin term for a plasma membrane was plasmalemma. So we call it a sarcolemma. Instead of calling it the cytoplasm, we would call it the sarcoplasm. Instead of calling it the smooth endoplasmic reticulum, we will call it the sarcoplasmic reticulum. And this way, anytime you hear those terms, you know you are talking about a muscle cell and not just a random cell in the body. Want to refresh your memory? The importance of the smooth endoplasmic reticulum is that calcium is stored in it, and that's going to be crucial for the functioning of skeletal muscle. And I also want you to realize that the sarcoplasmic reticulum is going to be surrounding each myofibril, which is going to be a group of contractile proteins. And I will be pointing this out on subsequent slides. So be looking for that. So let's look at this picture of a elongated skeletal muscle cell. Here is the plasma membrane, now called the sarcolemma. Notice that there are, like, if you took your finger and you pushed on the plasma membrane, you would have these little invaginations that would come down. Like if you took a piece of fabric and you pushed in on it. Okay, we're going to talk about those on the next slide. Then we have mitochondria. We're going to have lots of mitochondria in skeletal muscle because we need lots of ATP for that muscle to contract. Then this blue thing that looks like an old school radiator, that is the sarcoplasmic reticulum. This is showing the cut edge of it. You can see it's going all the way around these structures right here with this pale pink and those little dark red and blue dots. These are going to be those contractile proteins we will be learning about shortly. Okay, looking at another picture, just so you get used to looking at different pictures. Here, the sarcolemma is yellow. The sarcoplasmic reticulum is still this blue stuff, and the mitochondria, this time it's showing lots of mitochondria in orange. This one also shows the sarcoplasmic reticulum going around all these groups, all the myofibrils, inside of which you can see all the myofilaments. Okay. And so this is showing the little invagination, the little poke of the sarcolemma. And you can see there's lots of little places where we have this poking in of the sarcolemma. And wherever we have this poking in here of the yellow, that coordinates with this purple structure. And you can see this purple structure, which is going perpendicular to it or transverse. This is called a T-tubule, which just stands for a transverse tubule. And this T-tubule actually surrounds the myofibril, and then it goes around each myofibril. And so it's bringing part of the sarcolemma to surround each myofibril all the way deep within that myocyte, within that muscle cell. So It's a deep indentation of the sarcolemma, the plasma membrane. When it goes deep, notice it sits right next to the sarcoplasmic reticulum. And what's it being stored in that sarcoplasmic reticulum? Calcium. So that's going to be important. And it's connecting, it's surrounding every single myofibril in that muscle fiber, in that myocyte. Okay, so a myofibril is a group of myofilaments. Every myofilament is a protein. And so in these myofibrils, we have different types of myofilaments, and some are going to be contracting, some are going to be regulating. We're going to learn the names of 4 of them. 2 are going to be contracting, and then 2 are going to be attaching to one of those contracting ones. When you take anatomy, you may learn others as well. So here are muscle cells, and here is one muscle cell, one myocyte. Here is a myofibril coming from that. When that myofibril comes out, you can see the alternating, you can see the striation, the dark to the light, the dark to the light, the dark to the light. And so if we look at it on an electron microscope, you can see the light to the dark, to light to the dark. Okay. Here is another one showing the cell, nucleus pushed off to the side. Here are the myofibrils. There's a myofibril, another one, another one, another one. You can see how the sarcoplasmic reticulum surrounds each myofibril. You can see how the T-tubule surrounds each myofibril as well. Okay, and then you can see we're going to have a dark band, which is going to be here, and we're going to have a light band as well. So we have names for the alternating dark and the light. The dark band, take the vowel in the word dark, which is an A, is called the A band. So when over here in the colored picture is called an A band, that correlates to this part right here, which was the dark band. The light band over here, we're going to take the vowel in the word light, which is an I, and we're going to call it an I band. So on the electron microscope black and white picture, this light band is going to correlate with this I band. Now notice in the middle of the I band, there is this dark line. That is the same thing as this zigzag line shown in the picture on the right, and that is called the Z line. So the Z line is in the middle of the light band or the I band. Okay. Now, if you go from one Z line, which is in the middle of an I band, to the next Z line in the middle of the next I band, that is the structural and functional unit of skeletal muscle, and that is a sarcomere. Now, when you are writing, it's S-A-R-C-O, not like sacrum, which was S-A-C-R-O. So it's sarcomere. So a sarcomere is between 2 adjacent Z lines. So a sarcomere is going to have half of an I band, then it's going to have the next A band, and then it's going to have half of the next I band. And that is the structural and functional unit of skeletal muscle. Okay, so here's the quick think. Look at that picture. Now look at this picture. Can you find the A bands and the I bands on the light microscope picture? You should have been able to do that. Now in the middle of the I band, can you find the Z lines? You should have been able to do that. Now can you, with your finger, find sarcomeres? Okay, so make sure you're perfectly comfortable with those terms, because you need to know those terms going forward. Now we're going to add more terminology. Okay, we're going to look at that sarcomere. Oh, and by the way, here is another more powerful light microscope picture showing an I band with the Z line in the middle, an A band, and the next adjacent I band with the Z line in the middle. And I like this one because it shows a mitochondria here, and another one there, and another one there, and another one up here, so that you can see there's going to be lots of ATP generated so this muscle can contract. And so now we have the structural unit of skeletal muscle, and this is also the functional unit. This is where stuff is going to happen. So let's learn about the microanatomy of this sarcomere. And for that, we need to learn about a couple of contractile proteins, which are known as myofilaments. So filaments are proteins, myo in the muscle. So I can't draw that electron microscope picture. So we're going to go to a drawing that all of us can do, which are stick figure drawings. So all of you should be able to draw something like this. Draw your Z lines, then draw lines that are in the middle of the Z line and extend towards the middle of the sarcomere, but don't meet what are shown here in red. And then between the red lines, draw blue lines that do not reach to the Z lines. Okay. Now, the reason why this middle part is dark is because this part is very dense from all these overlapping proteins. And the reason why this part is light is because a lot more light can get through it because there's not nearly as much protein. So that's why we have the light and the dark. Okay. What I have drawn in red is a protein known as actin. So actin is always attached to the Z line and then extends towards the middle of the sarcomere on each side of it. Okay, so it doesn't just go to one side, it goes both sides. And then the thicker line in the middle of the sarcomere is known as the protein myosin. And notice that myosin overlaps the edges of the actin. Now we show myosin just kind of floating here, but if you take anatomy, you'll learn that myosin is actually attached to the Z line by something that looks like a little spring and elastic protein, but we're not going to learn about titin or any other proteins related to myosin in this course. If you took a human bio class or an anatomy and physiology class in high school, you may have got away with talking about thick filaments and thin filaments. Not for this class. You need to learn the terms actin and myosin. Do not write thick or thin for the answer for anything. I won't be using those terms. You need to know exactly what you are talking about. So let's have a little change in our thought process. Don't think this is too hard. I know we've done a lot of terminology. Instead of saying this too hard, just say, this might take me some time and some effort. After all, Dr. Powell and the dean told us day one, natural science classes, 3 hours for every unit. So we were looking, in addition to the lecture, we were looking for at least 6 hours of study time. No, sorry, 3 times 3 is 9. 9 hours of study time per week. Have I been putting in my 9 hours per week? If not, you need to start now. And you will find out that if you put in your 9 hours per week, you can do this. It won't be too hard. You will be understanding this. Okay. So let's go back and look at another drawing of those sarcomere bands, because I don't want you to memorize any one picture. I want you to be able to look at a picture and to recognize it. So now both the actin and the myosin are in green, okay? So the Z line is shown here in red. So we know actin is attached to the Z line, which makes this the myosin, okay? And so to tell the A band from the I band. The A band, by definition, is the full length of the myosin. So it goes from one end of the myosin clear over to the other end of the myosin. So if you're drawing a picture, do your dotted lines from one end of the myosin clear over to the other end of the myosin, and that's your A band. And then that will be your other A band. And so the I band is between the two. So notice it's not an I band for one sarcomere, and then here's the other I band for the other sarcomere. This is half of an I, and this is half of an I in each sarcomere. So to have an I band, you have to have two sarcomeres next to each other. And so by definition, what is an I band then? Well, is there any myosin in an I band? No. What is in an I band is the portion of actin that is not being overlapped by myosin. So what does that mean? Well, if we have actin and myosin overlapping, if they overlap more, is the A band length changing? Well, here, let me take two different things. If the yellow is myosin and here blue is actin, if they overlap more, has this length changed? Well, no. If they overlap less, has this length changed? No. So that's not changing. So the myosin, the A band length is never going to change. What is going to change is how much of the actin is not overlapped. So the I band length is what changes, not the A band length. All right. So myosin doesn't look like a highlighter. Myosin looks like a golf club. It has this long handle, what we call a bare area, and then it has a head. And what you do for myosin is you take like two golf clubs and you twist the bare areas together. And so you have two heads coming out on opposite sides. And then you take a whole group of these and you turn them so that the heads are going to come out in a whole 360, like that. And all their handles are in one direction. And then you take a whole other group of them and you turn them the other way. So here you can see all of these heads are this direction and the bare areas here. And then on these, all the heads are this direction and the bare areas are in the same area. So the bare areas are in the middle of the sarcomere and the heads are facing out toward the Z lines. And that's what myosin looks like. And that head is looking towards the other protein. It's looking to actin. It wants to stick its head into actin and interact with actin. But actin's got a surprise for it. And I put actin with a star behind it because actin isn't just actin. It's actin plus two other regulating proteins. Actin by itself is like a string of pearls. They're little ball-shaped proteins. And what you do is you take two of these little string of pearls and you twist them together. So that's what it looks like, these two little blue things. That's actin. But every little pearl has a hole drilled in it that myosin wants to stick its little head into. Little dastardly thing. But actin wears a chastity belt that covers that hole. So myosin can't get to it. And that chastity belt is a protein called tropomyosin. So tropomyosin is what I have shown here is this long orange protein covering the binding sites for myosin. The binding sites are in actin. Now, If we just had a chain of pearls and I just put a string across the top of it, what would happen? The string would hang down. So we have to have something to hold it into place. And the protein that holds it into place is a protein called troponin. And troponin is kind of like a Mercedes-Benz side. It's got three little prongs. One prong holds on to actin. The second prong holds the chastity belt tropomyosin onto actin to cover that binding site. And the third prong sticks up into the air, and what can bind to it is calcium. And we'll find out what happens to that in our next video, not this one. For now, it's time for you to practice. Study everything we've covered in this video, then come back, look what I've asked you to do without looking up answers, then go back, check your work for accuracy, watch it a second time if you need to, watch supplemental videos, talk with the tutor, talk with a classmate, come talk with me. I want to thank you so much for all your hard work. Remember, change your mindset. Don't say, I don't understand this. I don't get it. Change it to, what did I miss the first time? Obviously, I missed something. Let me go back to where I understood it and now figure out what I missed going forward, because I know you can do this. All right. I will see you when we come back to learn about how we're going to take all these pieces and let this little sarcomere contract.